Hello and welcome to Virtual Investor Conferences. On behalf of OTC Markets, we are very pleased you have joined us for our next live presentation from Sigyn Therapeutics. Sigyn Therapeutics are developing medical devices to treat cancer and infectious disease orders. Kindly note, attendees may submit questions and view our presenters' availability for one-on-one meetings by clicking Questions and Book Meeting in the top toolbar. At this point, I'm very pleased to welcome Jim Joyce, Chief Executive Officer and Inventor of Sigyn Therapeutics, which trades on the OTCQB venture market under the symbol SIGY. Welcome back, Jim.
Greg, I appreciate the introduction. Before getting started, I encourage everybody to review our forward-looking statements. We have also provided these on our website under the presentations button. That address is sigyntherapeutics.com. With that said, if you're not familiar with our company, we're developing next-generation blood purification therapies to address cancer and life-threatening infections. We designed our lead product to address infectious disease disorders that are not treatable with drugs. This includes sepsis, which is the leading cause of hospital deaths in the United States. Our closest clinical stage comparable to treat sepsis is a company whose market value is currently 20 times the present value of our company. Additionally, I previously founded a company whose infectious disease device drove a peak market value of approximately $200 million on NASDAQ during my tenure. I bring this up because much like our current trading status on the OTC, that's where we started with that company as well in our first few years. In regards to our pipeline, it's comprised of first-in-industry devices to improve the delivery of leading cancer therapies. Our pipeline consists of Sigyn therapy to address life-threatening infections, ChemoPure to reduce chemotherapy toxicity, ChemoPrep to optimize the delivery of chemotherapy, and ImmunePrep, which is a platform to optimize the delivery of immunotherapeutic antibodies. I will point out that chemotherapy is the most commonly delivered class of drugs to treat cancer. Additionally, ImmunePrep are the best-selling drugs to treat cancer in today's environment. Something you want to know about myself and my team: most of the members of our team previously worked with me when I oversaw the development of the first therapy to receive 2 FDA breakthrough device designations. This was the first device approved by FDA under emergency use authorization to treat viral pathogens. In this case, it was Ebola virus, and Time Magazine subsequently named the device a top 25 invention and a top 11 medical breakthrough. Today, we're advancing Sigyn therapy, which is a next-generation technology to not only address life-threatening viral pathogens, but to address a broad spectrum of other infectious disease disorders. And in this slide is a picture of Sigyn therapy in action during our first-in-mammal studies at the University of Michigan. When we designed Sigyn therapy, we wanted it to have unprecedented attributes. First off, we wanted to be able to have broad deployment in the case of commercialization. In this case, we designed the device to work on the infrastructure of dialysis and CRRT machines that are already located in hospitals and clinics worldwide. When we design an extracorporeal device, historically, these devices, to have success, to provide patient benefit, they need to have 2 factors to benefit patients. They need to be efficient. That means they have to have rapid turnover to process and treat the bloodstream on many occasions. Additionally, they have to have capacity to capture the targets that would be relevant in providing patient benefit. So in this regard, related to efficiency, our device is able to process the entire bloodstream approximately 15 times during a 4-hour treatment. And that's based on the average size of a patient. This is about 60% faster turnover of the blood processing or bloodstream as compared to the previous device I mentioned. Our device also incorporates a formulation of adsorbent components that have over 200,000 square meters of surface area on which to adsorb and bind our therapeutic targets. To give that some perspective, that's approximately 50 acres of absorption and surface area to capture targets in each cartridge. And then the last point, our device isolates viral pathogens, inflammatory cytokines, and bacterial toxins, including endotoxin, from the entire bloodstream. Now, the key word there is isolates, because previously there have been devices that concentrate pathogenic factors in the bloodstream. Our device is able to, on a rapid real-time basis, isolate our targets away out of the bloodstream, away from blood cells, where they discontinue any interaction with blood cells and where they can't have pathogenic factors lyse off that could cause or induce a life-threatening inflammatory condition itself. So this is a very important capability. As a result of these capabilities, our device has the ability or the potential capability to address several unmet needs in global health. Untreatable viral pathogens. You should know that a majority of viruses that are infectious to humans are not addressed with an antiviral drug. And when new viruses surface, the emergence of a new virus that's infectious to humans, these therapies, blood purification therapies, are the first line of defense. And I mentioned previously, our product was approved to treat, at my previous company, to treat Ebola. It's interesting to recognize that at COVID-19, the first 4 therapies approved under emergency use authorization to treat COVID-19 were all blood purification therapies. And so when these types of events occur, there's oftentimes a great amount of focus put on them because there's no other treatment options. An additional therapeutic target that continues to be an emerging global health threat is antibiotic-resistant bacteria. Sepsis, as I mentioned earlier, leading cause of hospital deaths in the United States, and endotoxemia. Endotoxin is a gram-negative bacterial toxin that is a leading promoter of sepsis. And in this case, we're addressing endotoxemia in end-stage renal disease patients. This provides us an early clinical and commercialization opportunity. There are more than 550,000 dialysis-dependent end-stage renal disease patients in the United States that receive over 85 million dialysis treatments each year. Once these patients are dependent on dialysis, their annual mortality rate is approximately 20%. That means the average lifespan is about 5 years for each individual. And endotoxemia and concurrent inflammation underlie early causes of death, which essentially are cardiovascular disease and sepsis as the 2 leading causes. The incidence of endotoxemia in ESRD patients is 60 to 90%. So that means the patient population opportunity is at least 330,000 individuals. Now, before I get into the clinical design of our first in human studies, I want to talk about the potential value of Sigyn therapy to the dialysis industry. And this is a real differentiator for not only this product, but the other products we create, because historically in the extracorporeal device industry, devices have been designed in advance that have been demonstrated to be life-saving. But in most cases, these products never impact an established industry. And so companies have to rely on marketing these products themselves, which also, in many cases, doesn't result in a favorable outcome. We recognize that our devices need to provide a strategic competitive advantage to an established industry. They need to affect somebody's business model. So in that regard, we see Sigyn therapy, if it demonstrates the ability to extend ESRD patient lives, that increases revenues of the dialysis industry players. And this is quantifiable and it's compelling. It also provides these companies a pathway into very significant non-dialysis markets, and we'll talk about that in a bit. In North America, the industry is dominated by 2 players. It's Fresenius and DaVita Health. DaVita has over 200,000 patients in their North American network, and based on average annual revenues per patient, extending the lives of these patients by 1 month, for each month you could do that, that adds $960 million in increased revenues. ESRD patients are also in the hospital quite a bit each year, upwards of 2 to 3 weeks. And for each week of reduced hospitalization, that would allow DaVita to recoup approximately $240 million in revenues. For Fresenius, based on their patient population, each month of extended life for their patients would add $1.2 billion in revenues, and each week of reduced hospitalization will allow for recouped revenues of approximately $300 million. Our clinical plan for treating the ESRD patients, we've drafted an investigational device exemption for FDA submission to initiate a study that will enroll 12 to 15 ESRD subjects with endotoxemia, concurrent inflammation at site locations in Hartford, Connecticut, San Antonio, Texas, and Las Vegas, Nevada. One of the things that we're preparing to do in the coming weeks is to submit a breakthrough device designation for this indication. We've had success previously with breakthrough device designations. We have not yet submitted one for Sigyn Therapeutics, but we're optimistic. There's no assurance, but if that is awarded, that will allow us to fast track our communications with FDA related to the IDE we're planning to submit. In clinical practice, our device will be administered during regularly scheduled dialysis treatments. So these patients are in the clinic 3 times a week, 4 hours of treatment. Nothing changes for the patient. We put our cartridge in series with their dialysis treatment, and the clinical protocol is structured in a manner that actually provides us valuable data that you normally can't find or obtain in what's known as a feasibility or safety study. So in this case, there's 6 treatments over the course of 2 weeks. Treatments 1 through 3, it's the dialysis patient receiving the normal dialysis treatment. We measure changes in blood chemistry, including reductions or changes of endotoxemia or pro-inflammatory cytokines. And then in week 2, we add our cartridge in series during treatment 4, 5, and 6. We look at those same changes in blood chemistry, and what we hope we'll see is reductions of endotoxemia and pro-inflammatory cytokines as compared to dialysis alone. Setting the successful completion of this study will tee up what's called pivotal studies, which for a class 3 medical device, this is the study that's required for market approval in the indication. But we're designing this as a modular study, where this study serves as a safety study that allows us to bridge not just into ESRD applications, but to also bridge into non-ESRD applications. And this is where sepsis comes in. We have an opportunity to move into sepsis studies if we're successful with this study. Something about sepsis, as I previously mentioned, leading cause of death in U.S. hospitals. It costs the U.S. healthcare system approximately $38 billion each year. It's a dysregulated immune response that in most cases is induced by either a bacterial or viral infection. This results in many cases in an excessive production and pro-inflammatory cytokines. When their production is dysregulated, a phenomenon occurs which is known as the cytokine storm. Today, we know that a big inducer of this are bacterial toxins that leak from the gut. So as a result of infection and inflammation, that creates permeability in the intestinal lining, which allows resident bacterial toxins in the gut to leak into the bloodstream, which can cause a potent activation of the immune system, which can lead to organ failure and death. In regards to our sepsis-related studies so far, we've demonstrated the ability to clear viral pathogens from human blood plasma. We've demonstrated the ability to clear relevant pro-inflammatory cytokines, TNF-alpha, interleukin-1 beta, IL-6. And then we've demonstrated the ability to eliminate bacterial toxins, including peptidoglycan, lipoteichoic acid, and endotoxin. And I'll point out, this is a critical collection of data because sepsis is a multifactorial condition. At present, and this is something you want to keep a close eye on, the current clinical stage device to treat sepsis in clinical studies that's being advanced is the polymyxin B device, which is solely limited to removing endotoxin. And this is being advanced by a company called Spectral Medical in clinical studies here in the US. And this is endotoxin-driven septic shock. And I would encourage you to follow this company, because what Spectral has done is they've developed an assay that's now FDA approved that helps guide the patients that they treat. And what they've discovered during previous clinical studies is that when there's excessive levels of endotoxin in the bloodstream, their device does not provide clinical benefit. It does not reduce mortality. If the levels are too low, this product does not have a significant benefit. But when they enroll patients within a certain range of endotoxin, it seems as if the product provides benefit. And we believe there's a possibility that at some point in time during the first half of this year, that they may complete the clinical study and potentially receive approval from FDA, which would be very, very beneficial to us based on our ability to address endotoxin plus other bacterial toxins in combination with pro-inflammatory cytokines. And if the sepsis was induced by a viral pathogen, the ability to potentially dampen down the pathogen source of the inflammation to begin with. In oncology, I want to point out that we don't develop drugs to treat cancer. We're developing devices to improve the benefit of drugs to treat cancer. First, I'm going to talk about chemotherapy. As mentioned at the outset, most common class of drug to treat cancer. You may be surprised to learn this, but less than 3.5% of the drug actually gets to the tumor cell targets. That means the other 96.5% is off target. And off target chemotherapy is more likely to contribute to toxicity than patient benefit. And a problem with off-target chemotherapy is a lot of it is getting bound up by particles released by tumors. These are tumor-derived exosomes. We'll call them chemosomes in this application. But these particles suppress the immune system of cancer patients. They play a role in seeding the spread of metastases in cancer patients, and they are in immense concentrations, and their quantity and circulation correlates with disease progression. As it relates to chemotherapy, they are directly associated with chemoresistance. They can interfere with the delivery and also extract chemotherapy back out of tumor cells after it has been successfully delivered. So our solution is 2 steps. Chemo prep is a pretreatment depletion of chemosomes to improve the delivery of chemotherapy to cancer cell targets. We envision that this system will be an easy to deliver system. It'll be in the infusion centers where patients are receiving their chemotherapy infusion, and it'll be treated prior to that infusion to reduce the circulating burden of chemosomes. And we believe that will improve delivery. And our goal is to be able to maintain effectiveness of chemotherapy with lower doses. And this is critical for 2 reasons. If you can conduct or maintain efficacy with half a dose, well, that cuts right up front, that cuts toxicity in half. Additionally, there's been global supply chain issues with chemotherapy, which in the US has caused rationing, and in many cases, patients have not received their chemotherapy and subsequently died. We believe that this could help potential supply chain issues. And the other aspect is there's an initiative with FDA called Project Optimus that actually is seeking for therapies to maintain benefit of cancer patients with lower doses. After this device, after chemo has been delivered, we then have chemo prep, which clears out the remaining chemotherapy left in circulation that is unlikely to cause benefit at that point. We do that to further mitigate treatment toxicity. In regards to immunotherapeutic antibodies, you're probably familiar with Keytruda, which I believe did a little over $25 billion in revenues last year. These are the top selling drugs to treat cancer. And these are the most valued assets in global medicine based on 2023 and 2024 M&A activity. You might recall Seattle-based Sigyn was acquired by Merck for $43 billion. And then there was the Pfizer acquisition of Prometheus which was a clinical stage company, no approved antibody assets, and that was a $10.8 billion acquisition. There's over 1,000 human studies evaluating therapeutic antibodies, but yet there's no strategies to improve their delivery. And the limitation of these therapies is that less than or approximately 2% of doses actually reach their intended targets. And so if you consider that, that's bad economics. $10,000 drug regimen providing $200 of patient benefit. Additionally, these drugs are known to be part of what's an emergence of precision medicine. Well, the fact is they're very imprecise because while they're antibodies that can have an exquisite specificity to bind to a protein that's on the cancer target, this protein can be, which is the receptor for the antibody, can be circulating freely, and it can be on many other different particles. And so in this case, these are decoys that display the receptor site that remove the antibody before it can get to its intended designation. And as a result, most people don't respond to these therapies. In our case, Immune Prep is a platform to incorporate therapeutic antibodies within a cartridge to sweep decoys from circulation prior to the subsequent infusion of the same antibody into a cancer patient. And I sat in meetings I always recall sitting in meetings at CDRH, which is the medical device division of FDA. And I recall a meeting where they said, Jim, if you had only incorporated an approved biological agent in the device, we could give you this accelerated pathway towards market. So in this case, that's what we're doing. And we don't only anticipate that we could improve the delivery by removing these decoys, but we could simultaneously clear the intended target of the antibodies to further enhance patient benefit without added drug toxicity. And again, how do we create strategic value that would make us important to an established industry player? We believe by expanding the use of therapeutic antibodies and enhancing their benefit would provide strategic value to the drug industry and would provide a new intellectual property pathway that could be very valuable to the pharmaceutical industry. So in conclusion, we're developing next generation blood purification therapies to address cancer and life-threatening infections. Our lead product is positioned to address infectious disease disorders that are not treatable with drugs. Our early opportunity is to treat ESRD patients suffering from endotoxemia and concurrent inflammation. And that allows us a potential opportunity to bridge into treating sepsis. And our pipeline is comprised of first-in-industry devices to improve the delivery of leading cancer therapies. So we're a public company. Let me give you some background data. You can see our 52-week range. Our shares outstanding, 1,605,377 shares. What you should know is greater than 90% of our shares outstanding are held by management, 2 investment funds, and 1 investment group that continues to build their position in the company and have been immensely supportive. Our finance and marketing plan, we've kicked off a Reg D private placement. It's a 10% convertible note. We kicked this off last month and are making progress with it. And this will fund the initiation of the ESRD patient study. By the end of Q1, we're launching a media and investor relations program to introduce our company to a broader universe of investors, and also to seed the launch in Q2 of a Regulation A uplist to NASDAQ offering, where the offering will be open for up to 1 year to raise a maximum of $20 million. And our goal is to meet the shareholders' equity requirement that would allow us to list on NASDAQ through this offering. We have an application on file with NASDAQ, but there's no assurance at this point in time that we'll achieve that goal. But we do believe people, as they learn about our company, should be very responsive to what we're doing. At the top right-hand corner, of this slide. Offering memorandum, if you want one, contact Johan Lowe, and I'm going to leave his number there and his email address. Johan recently joined our company as Director of Strategic Programs. Prior to joining us, he held a similar position at another extracorporeal company where he raised more than $30 million for the company. So it's nice to have Johan on the team. So in closing, here's my contact information, and let's look at some questions. We have one question. What are key milestones for 2025? Two key financing milestones, as previously mentioned, completion of our Reg D private placement, and then the launch of our Reg A broadly marketed offering to the financial community and to both accredited investors and smaller investors. The proceeds from our Reg D will launch our study in ESRD patients, and parallel to this, we'll be kicking off proof of principle studies, in vitro studies with our other products in cancer. One is, do we have any agreements with dialysis centers? I will say that we know the two leading companies in the dialysis industry quite well. We have worked with one of those companies to develop our clinical protocol, to provide us our clinical site locations and our principal investigators. So, we believe that the successful completion of our feasibility study, will, if we can demonstrate safety and demonstrate that we can reduce our targets, endotoxin and pro-inflammatory cytokines, then that will put us in a very advantageous position in extending our relationships with the dialysis industry. And again, we know the industry well. You'll see some of our advisors come from the industry and have leadership roles. So we think we have a good strategy. One question is, are slides available? Yes, we'll have these up on our website. Again, that's SigynTherapeutics.com. And they should be up now under the presentations button. Next question. Can you go through the difference between your product and that of the public company I previously discussed? Yes, our product is a adsorbent-based device it's a larger device. It has faster flow rates, greater capacity to capture a wide range of targets, both infectious viral targets, pro-inflammatory targets, and bacterial toxin targets, whereas our previous device was incorporated a biological agent that bound to a high mannose glycosylated structure that's on glycoproteins that coat viruses. And so that gave us broad spectrum capability to address viral pathogens. But today we know that it's not just the pathogens itself, it's the downstream effect of the pathogen creating an excessive inflammatory response and the translocation of these bacterial toxins from the gut into the bloodstream, which can then cause a dysregulated response that leads to organ failure and can cause death. Okay, well, we did a good job. That's all the questions. We're at 29 minutes and 18 seconds. So I appreciate everybody's time, and I hope you follow our company closely. Again, I'll go back a couple slides. If you're interested in the offering, I will leave this up. You can contact Johan, and there's his contact information. With that, I thank